You probably knew from what I was telling you earlier that there has to be some genetic code. There has to be some way of achieving this process of translation. We talked about it when we talked about making the protein. It was implicit in there. It's even implicit in the name translation. I mentioned earlier that there were 4 bases in mRNA, A, C, G, U, but 20 amino acids. That doesn't simply track. I can't use the base A to represent a particular amino acid because I'd rapidly run out of amino acids to code for. So it should stand to reason that if I take 4 bases, one at a time, A, C, G, U, that I can code for 4 amino acids. That's not enough. If I take them 2 at a time, AA, AC, AG, AU, CA, CC, CG, CU, and so forth, then I can code for 16 amino acids. That turns out to be 4 times 4, or 4 squared. So if I take them 2 at a time, and I have 4 bases, then I have 4 squared possible combinations. If I take them 3 at a time, I have 4 times 4 times 4, or 4 cubed, 4 to the 3rd possible combinations. That allows me to code for 64 amino acids. So 3 is the minimum number of bases that can code for 20 amino acids. And that's why the codons that we looked at in earlier objectives had 3 bases in them. AAA, AAC, AAG, and so forth. So here we see those 64 possible combinations. There's 64 combinations, which is more than enough to code for 20 amino acids. For that reason, we call the genetic code degenerate. And degenerate can mean a lot of different things, but in this context, what it means is that we have more information than we need. We have 64 different ways of representing this, but we only need 20. So we'll see the results of that degeneracy in the next couple of slides. There are 64 combinations coding for 20 different amino acids. The 64 combinations are shown at left. 20 amino acids that we studied in Module 3 are shown at right. As we said in an earlier objective, mRNA matches up to tRNA in order to create this protein primary structure. You saw the middle of this slide in an earlier slide that I showed you in Objective 10. Now we're adding information that we saw earlier, the genetic code, the 64 possible combinations, and we're adding in the primary structure of a polypeptide, which is going to turn into a protein, that we saw earlier in Module 3. So there's a series of peptide bonds that are holding these amino acids together in a primary sequence. So the way in which we carry out this translation is the cell's equivalent of a French to English dictionary or an English to French dictionary, a translating dictionary. It's called a codon table. And so the codon table is the code that we use to convert this mRNA message to an amino acid sequence. We saw earlier that AUG is a start codon. AUG codes for the amino acid methionine. There are 3 stop codons that cause the ribosome to be released from the mRNA. They're UAA, UAG, and UGA. And an example of something that may occur in between is the codon GGG, which codes for glycine. These 3 stop codons have names, not something that you need to learn. I just think it's an interesting point. The 3 names for the 3 stop codons are named after colors. They're called amber and ochre and umber. Well, why would we have 3 different color names for 3 different stop codons? Well, it turns out that a lot of this work was done in fruit flies, and in fruit flies that have the mutation amber, they have amber colored eyes. This is the same thing we saw earlier for beta globin in beta thalassemia. It's an abnormal stop codon. So where we have a mutation or a change in structure that results in an abnormal stop codon, it makes the eyes a different color in these fruit flies, an amber color, and so that's called the amber mutation. So it was a stop codon at that particular point. And just so you know what kind of geek I am, I'm enough of a geek that on this slide I went in and found the correct RGB codes, the correct color codes in computer language for amber, for ochre, and for umber. So those are the colors of the fruit fly's eyes. In the example we're showing here, you have a codon table. We're going to show how to use this codon table. We have the codon UGG, remember still 5' to 3'. It matches the anticodon CCA, still reading 5' to 3'. So UGG is the codon which codes for tryptophan. This is the same example that we used earlier.